So welcome to Astrobiology Colloquium. Today's speaker is, oh, you're going to get rid of that, yeah, is Fatma Pir Cakmak, currently at Grand Valley State University in Michigan. It's been about a year. And before that, she was a postdoc at MIT. And before that, she did her PhD at Penn State University. And she's invited because she's doing some really interesting work in prebiotic chemistry about phase separation and how that can enhance prebiotic reactions. And so she's going to tell us all about that today in her talk, Exploring the Emergence of Life, Phase Separation as a Means of Compartmentalization in Protozoan Models. So take it away, Fatma.
Hello, everyone. Thank you for this opportunity. I'm very excited to be here. Today, I'm going to talk about how life might get started. Earlier, I'm going to specifically focus on compartmentalization strategies for protocell models. Some of the intriguing questions that motivated my research includes what's the difference between living and non-living matter? How could we assemble from simple probiotic molecules a protocell? So what fundamental chemistry underlies protocell models and living cells? And what do they have in common? So I used separated systems. Here you can see here as beautiful droplets and lipids to tackle this question. Okay. Let me actually bring that back. Okay, sorry about that. Now I think people can see me for the recording as well. Let me also hide this. Perfect. So before start, I would like to actually describe what I mean by a protocell, right? Because there are many definitions. And for this talk, when I refer to a protocell, I will refer to a protocell as a compartment, self-assembled compartment, aiming at explaining how more complex biological cells have evolved. So I know many of you actually learned like this aspect as well. We actually met today. Please forgive me, I will do very broad definitions and very broad terms. I will just mention things very broadly. If you look at here, we're going to see a very simple frame, right? We actually had Earth formation around 4.5 million years ago. And it took here to serve Earth's life. We are talking about a billion years. There are many different scenarios to explain how life might have started. So, but in very, very broad terms, I cannot emphasize the broad here. You have a fluid chemistry, right? That leads more complex biochemicals and subsequently evolution of life. Okay. So some of the examples of these theories includes showing the third Miller-Urey experiment. This experiment showed that you can produce various amino acids easily in a simple container like this. Here it's Daniel Miller pictured during the experiment in San Diego. He has very simple chemicals as a starting material, and you have reducing mixtures and electrical charge in this reaction. And as a result of this experiment, they were able to show that they can produce different type of amino acids. So another experimental evidence that you might have some biochemicals available is people actually look at meteorites meteorite extracts, specifically, not just in meteorite. And they see that from the extract, they were able to form vesicles. You have this simple, relatively simple molecules, right? They might exist from different theories possible. And you might ask how long the polymer might get, right? And another scientist at Paris showed that using simple minerals, display the light of monochrome, they were able to oligomerize the simple molecules and organisms 55 monomers length, and which is quite interesting. So this is just the introduction. So now you have this primordial soup. You have this amino precursors, nuclear precursors, and the next question is how the first cells assemble from this components. Here, you're going to see a vesicle, right? If you have liquid precursors, you will have a group that will be hydrophilic, and you will have a tail group that will be hydrophobic. So here you have a facility, and they have the same properties, hydrophilic head, hydrophilic tail. If you put these molecules in water, what will happen is they self-assemble into these vesicles. They will form this compartment instantaneously. Other way that we can actually form compartments is going to be place separated droplets. Again, from amino precursors and nucleotides, you might actually, these molecules can come together. Instantaneously, they can form these droplets, biomolecular bond space, and dilute continuum space instantaneously. I will talk about this later in biology class. So, you might say, okay, why do you care about compartments? Why compartments are important? This is actually a picture from my hometown. I'm from Mediterranean, so Mediterranean Sea. This is a sea, right? If you have this very precious biomolecules, what will happen in ocean, right? They will just degrade. Nothing interesting will likely to happen there. However, if you have a compartment, right, like cell itself, it's a compartment, you can have this biomolecule concentrated in this compartment, and you can have that important biochemical reactions that can exist in this little compartment, because it will be possible for this biomolecule to interact. Okay, as I said, cell itself is a compartment, and it also uses membrane bond and membranous compartments to further compartmentalize biomolecules and to regulate reactions. It is necessary for life. And there are other things like compartmentalization can help you, or you can maintain high local concentration. You can have selective entry and exit, and you can also provide favorable condition that's different than the outside. Right? So for this talk, I'm going to focus on compartmentalization strategies, specifically membrane bond and place-separated droplets. And first, I will focus on place-separated droplets. You might think that you actually understand how vesicles work. Right? Cell has a membrane. It's easy. I understand why you learn from there. How about place-separated? Actually, place separation also takes place in extant cells as well. Activities in cells can be place separation. Can be compartmentalized by furthering cells by place separation. There are a number of cytoplasmic and nucleoplasmic cellular condensates, RNA and protein are reported to have nucleotide characteristic. What is this characteristic? Yes, it's spherical shape. You can have dripping infusion, right? And this form nucleolide from sides, and you have people from C. elegans. And these two heads will have very important consequences, such as you can have biomolecular partitioning, you can have co-compensation, and sequestration, and so on. And people actually, this number of cellular condensates that has been discovered is on the rise. This is just from 2017. There's actually an updated version for this. It's not published, but it's in the first author to show in a conference. The updated version. It's getting the number of comments that they discovered getting higher and higher. Okay. Even though the phase separation in South is a relatively new concept, phase separation existed almost 100 years ago. The one type of phase separation I'm going to focus on today is going to be complex conservation. The first time the term coined is 1929. It was coined by Deon and Coach, and they told in their paper an unmixed formation of flux, which may even manifest itself as separation in the two liquid layers. And this kind of theory is going to look like very similar to the pictures I will show you. So they took place almost 100 years ago. Okay, I mentioned phase separation that exists before, and I'm not the first one to claim phase separation might involve in organic life. There's actually a paper, it's a book about it by Alexander Okhrin. He's a Soviet chemist, and he said that life might have evolved from causal rates, and lipids, right, they're called by electrolytic forces, and he claimed this in 1936. I think it might not get the first solution. Some of the tourism might play a role, but this actually has been claimed in 1936. So, what is the phase separation I'm going to talk about? How does it exist? What's the modern? So, when you have anything charge, it's the nucleotides, polymers, anything you can immediately charge. If you just combine this all the time with the positive charge molecule, the negative charge molecule, if you put them at the right ratio, you're going to form condensed positive phase and direct continuous phase. They will exist with equilibrium and just you're going to just mix them. You're not going to use any special thing. You're just going to mix the solutions at right conditions, and you will form separate systems. So written here, we can discuss in more details. We will like to talk about it, but to tell you there are different type of phase separation and the driving force for each integration. So, what does it look like? When you are in the lab, you're just mixing those like polar electrolyte solutions. Let's say you have negative charge and positive charge. You mix them, nothing happens. You will have a water liquid. You're not likely to observe any droplets. You mix them, and you have high turbidity, like here in the container microcentrifuge that you have here. You see this cloudy solution forming, right? It seems it's an indication that that so probably won't droplets. You can just UV this to differentiate between those. It's not always easy to differentiate that phase by your eyes. I'm just showing extreme examples. Sometimes it's always useful to have a usually differentiate. So, as I said, if you have a clear solution, you will likely just have a solution under the microscope. You're gonna likely just see nothing, just polarized holding for a solution, right? Not any detectable structures. But if you have a turbid solution, you can either have causalary droplets or aggregates. You need to use a microscope to differentiate between this. So, how can you differentiate? If you have a causalary, you're going to have fully spherical structures like here. If you have aggregates, you're going to have amorphous clusters. Okay. Let me tell you, this is just not one or the other. You can actually draw between those structures by just simply changing the polar concentration, then charge, density, and ionic strength. So I really like this example right here. It shows that we have an aggregate, right? And for aggregate, you can go to solution formation by simply changing the concentration of salt. Right? You have no salt here. You actually have this electrolyte mixture. It's forming this aggregate. Right? Increasing the salt concentration, you can move from the aggregate to chloride formation, like the phase separation right here. If you increase the salt further, you're going to have this homogeneous chloride formation. This is just an example that one property can change. How one property can change the transition to those concentrations. Okay. You might actually ask me, how can you use collaboration? Collaboration is very helpful in removing liquid organelles and extinct cell and protocell models. I don't see them as separate, right? The fundamental physical chemistry of the ship. Today, I'm going to tell you two main stories. There are going to be side stories as well, but I'm going to mention two main stories. First, compartments. What's the function of this compartments, right? Especially face-to-face. What can you do with those? What is the advantages? And the next one is I'm going to use the template, the concept template, giant physical formation. I'm going to start with the function of the families for Gamma's compartments. Let's say you have a polar system. You have the polio here, which is basically very long mu, RNA, and heat spur. If you mix them, you have your term solution like that. If you just slightly bit, by the way, you're using very, very low concentrations, 0.1 big percentage here, by the way. Very, very good level. And if you slightly bit this term solution, you will see the color phase with its tiny face. It's very small. But that's not. If you look at them in this, if you see some sample on the microscope slide, you look at under the microscope, it's something like this. So, when you have the color rates, then you add extra labeled RNA, in this case, labeled red. If you add any biomolecule, it's going to partition selectively into droplets. By the way, the amount of RNA I made is very, very low. Very low. So if you look at here, right, you have the closure rate sample, you're adding the RNA that gets concentrated in the droplet. That's why you can see the fluorescence. Otherwise, there will be some RNA here, so the concentration yet. It's not possible to fluorescence. If it was equal to lot, you would just see red, but we don't, right? So they get concentrated in the droplets. You can calculate the concentration in the droplets by just simple using calibration curve, and you can get the concentration, how much concentration you have. And that's what I exactly I did. So we have the closure. Well, okay, nothing is returned. Awesome. So I have these three different type of RNA. The first one is I have U15. Uridine, I have uridine that's have 15 nucleotides of uridine, and the M15 means I have random nucleotides. It's still they are all the same length, all 15, but the identity is different. The first one is uridine, second one is just random nucleotides. You can have anything, and A15 is just completely patterns. So I check the partition property. So we have droplets, right? How much of the things goes to droplets? I measure the concentration in the droplet, and I divide it by the concentration of the terminators. Right? That gives you the partition coefficient. That means higher the partition coefficient, higher the amount of molecular RNA that will go to the droplet. So you have for U15, and your here the causalary system I have is polyunsaturated. It has the biggest binding solvent, right? And for A15, it has the strongest binding peak polyene, and this is base rate. We get the numbers, right? It also reflects it. For A15, we have a partition coefficient almost 3,000. And if you look at field dependence, the smallest 3, it's around 44 times. It's only 44. And it's a high number, you can still concentrate things, but when you have a specific instructions, you can concentrate things more than the others. So there is one way you can actually use this closure risk reflex. Another way is like I decided to check, right? The length is very important. Identity of the molecule is important. I decided to just check the biological relevant molecules that I can use and how they will work out, and how short of the molecules I can use with closure risk. Since in order to make a closure rate, if you have a higher charge density, if you have a longer molecule, it will have higher capacity to place it with. So my question was, how low of concentration I can use and how short of the molecule I can go. And for that, I had different type of molecules, and I used different lengths. I changed the length from 1 molar to 100 molar, and I had positively charged amino acids and negatively charged amino acids, and I changed the length for every single one of them. And I also decided to use nucleotide. I used RNA, ATPA, between AMP, different number of phosphates, so that does change the number of phosphates you have, and hence the charge. So I also, we actually discussed the conditions we will use for this type of experiments. We decided this low buffer concentrations might be an example of a cone condition that we can use. We use this buffer concentration, and I also use only 10 millimolar charge concentration. Charge concentration, when I say charge concentration, charge of molecule times concentration of the molecules will give you the charge concentration. It's 10 millimolar. If you have 10 charge, that means you only have 1 millimolar more. So, and I start making the phase diagrams. When I check all of these positions, what I observed was very interesting. If you have arginine, you can just form closure rates. Let me explain what you see in the phase diagram. If you have no formation, you're going to see this empty sphere like this. It means no formation. If you have a closure rate, you will have this red circle. When you have a closure rate, you're going to see that. When you have aggregate, when you mix those things at this condition, you're going to see this like dot. So if you look at this table, right? For arginine, you can form closure rates with AMP, which is normally the speed of that rate, and it's 10 molar arginine. And you can actually go on and have 10 molar with one closure rate. The same thing for lysine. You can actually go and form a 10 molar lysine. This is actually targeted length. This is identity of the nucleotide table. You have lysine here, monomer, 5 molar, 10 molar, 30 molar, and 100 molar. The same thing for all of the tables you're going to see. So, and this is how you're going to do the clean for the tables. I thought it was quite interesting, right? You can actually go that low for this structures. And the pictures represent specifically from this graph. You have the arginine, no formation. You have the polymer, this forms like this. And you have the rates again with first step. And I was actually just have all this table, all of this condition. And I have seen some trends, I observed some trends. For instance, protein plus seems to form more aggregates compared to aspartic acid. And arginine seems to form closure relation with shorter length of lysine. 5 molar of arginine can form closure relation. So these are the tables. They are very interesting, right? How it actually shows you how low you can go. And then we decided to further analysis of this closure rate systems. We had chosen different lengths, and we were going to try how RNA partitioning is going to work out in these different systems. And I actually decided to one more system with ATP, since ATP is interesting molecule, and see what happens. This is the partitioning. So when you have the covalent system, you have, and I had single-stranded RNA 10-mer and 20-mer. So in different systems, you will see that, except this one right here. You know that for the sake of time, I will explain it later. But basically, what's here that when you have a shorter compartments, it's adding, by the way, 0.1 micromolar into the solution, and they can get concentrated 40 micromolar and more for the shorter compartment. Here we have this little star. So I also observed this little tiny, maybe it's a second phase formation. We didn't analyze, but with the microscope, you have a certain resolution, right? It was very interesting. So I actually have here a trend going on, right? For the shorter ones, it's partitions better, and for the compared to the longer ones. Okay, the next question we asked was, how about the structure? What happens to the structure in this practice? And in order to study the structure, I actually applied that. With this synthesis energy transfer mechanism, I look at double-stranded RNA. So whether so if you have a double, if you have same breakfast, and I will explain this in diagram. So when you have two molecules, right, if they are close, there's energy transfer, and you will have higher effect signal. If the molecules are far apart, you're not going to be able to have energy transfer between them, and you will have a lower so when they are close, you get the signal. If they are apart, you're not going to get high signal. And it can report to 10 nanometers within single. So for the system I have, right, so I have a closure, I use double-stranded RNA. So if you have a complementary sequence, if you are actually forming a double-stranded structure, they're going to be close, and you're going to have high pressing. If they're apart, right, more than 10 nanometers, you're not going to have to. This is actually just a control experiment, right? You have a zero threat signal as expected because these are the same strand. The same strand is not going to form a double-stranded structure. So I actually have another control. This is a buffer. This time I have here is a single-stranded stuff, and anything I will show here will include the double-stranded. All right. So here is going to be the high signal we can observe. Right? We have the double-stranded RNA in the buffer, right? That's going to be the highest signal you will observe because double-stranded structure is not going to unwind. It's just a buffer. So when I add this double-stranded structure in different causaries in different condensates, I actually observe that the thread signal that decreases as you increase the length. This is quite interesting, right? For the smaller quantity, that means you can preserve the double-stranded structure, but then you increase the length, right? As you go from here into there, you increase the number of what would you have? It seemed like the structure was destabilizing, it was unwinding. So, here to summarize, you have shorter focus time with polyamides right here. And if you add RNA, it's going to preserve the double-stranded structure. If you have more complexes, it's going to destabilize, it's going to unwind, delicate the RNA structure. This has quite different consequences. It is actually very interesting in terms of origin of life. Actually, I will try to show you that the shorter counterparts, with small molecules, are better concentrating the RNA compared to the longer parts. And the shorter compartments can also accumulate the function molecules than preserve the structure. Structure is very important for function. So you can actually transfer the base pair information, and you can also transfer the signal structure for coupled data. Your previous slide. It's not possible because of how like they're not going to be close enough in terms of I tried many different control experiments. For instance, I mean, you have the highest rate you get, but they have to be close enough 10 nanometers. But 10 nanometers is the actually couple. So they have to be closer. And I actually tried when you have the single strand structure in the quasar rate as an experiment. If you had like the same thread, whether you get any thread signal out of that, and you don't. I don't show the results here, but there's still like not very concentrated quite significantly. Does it answer the question? Okay, cool. Is there any questions I can answer? Okay, so this was the functional aspect of quasar. And I'm going to move on how we can combine two different compartmentalization methods and get some interesting results. So I'm going to focus on this type of vesicle formation. As I mentioned, right, when you have a vesicle, then you have a lipid molecule, you have hydrophobic tails, and you have hydrophobic heads, amplified. Then you put them in water. They will sample into vesicles for the structures, and you can use compartments. And there are definitely advantages of just having vesicles versus just space-separated systems. There are also advantages having the space-separated systems versus the space. Okay, there's also interesting papers about this in the field. People are trying to show that you can have phase separation and basically bilayer formation around those vesicles. It's quite difficult to form bilayer around the full electrolytes because vesicles themselves are quite patchy. There are no surface charts, and you also have another chart, and you're trying to just make them behave together. That's quite difficult. So people did some experiments that just triggered the charge change in the structure, and they're going to form like here you can see the when they change the pH. But there weren't any papers at the time that was able to form the bilayer around the proteins. So why do I care? Why do I want to combine this? So since you have a bracket, right? And you can call it bracket, you can actually do interesting stuff. So if you put the self-assembly recipe, you can have a microreactor here, and you can have form type of restriction, you can form reactions within this. And then you can actually form, you can use the polymer space templates, giant vegetable around, you can form a bilayer. There are different things you can do. So what is a bioreactor? I also did some experiments early in my PhD. I used phase-separated systems, and I stabilized one of the phases using clay particles, and I used this as a catalyst for a reaction. And I also did the effects to show it's quite interesting to use this as a bioreactor. So here, you can actually form self-assembly vesicles around the protein, and you can also form bilayer around the protein. How are you going to differentiate between those? And can you do it directly, or do you have to prepare that using different methods? So, in order to differentiate, first, these are actually micro-size structures, right? How are you going to know what you have? You are going to actually look at the diffusion coefficient. So, if you have a bilayer, it's structure, right? You have a bilayer around it. If you have a diffusion here, if you're you can actually tell that you have And there are other experiments to confirm that, but basically you can tell whether you're a bilayer or not. If you don't have diffusion, you have just difficult to see it. And for the diffusion, I use person's recovery after the diffusion. Then you see the extra data. You will see a graph like this. You actually have first graphic of interest or region of interest. Before the region, you have the fluorescence, you reach the region, and then you monitor the recovery afterwards. And you will get something like a graph like this. So since the lipid molecules, if you have continuous layer, they will move and you will see diffusion. If you have this one here, you're not going to see any diffusion. It will be immobile, you will see no recovery. And here you can see mobile, you will see plus recovery. I'm not going to go into details, but I was able to plot microreactors like this again. This doesn't impede any diffusion in RNA molecules. You can concentrate things after, you can add those into these droplets, and you can form this assembly like this. And after this, I was really determined to form a bilayer around causaries. It was a challenge, but still, I was very interested. So we find a method that could actually help us to form this. There's a general method people usually prepare their lipids. It's a gentle hydration method. What they do is that they have a dry lipid in a glass container, and then they dry this lipid and they put their hydration solution, whatever the solution they want, or whatever they think they want to encapsulate. And then after they let like 1 day in oven with hydrate solution, and they will form this vesicles from here. This is the just general method dates. So then you form a vesicle from here. You're going to see the transit part in the fluorescence. Vesicles are going to be global intact, and you're going to see the lipid location by red. Okay. So then you actually form vesicles using this method, the hydration method. You have very heterogeneous samples. You are going to have some vesicles that you can study, but it is difficult to get homogeneous samples. So instead of using just a regular phosphorus solution, we decide to use closure rate and see what happens. You have to have the right closure rate and put a combination for this. And then we add the closure rate instead of just buffer. We were able to spontaneously form bilayer around it. We just hydrate the solution, we just use hydrating solution, and we were able to form a bilayer around the participants. Here, we observe that all there actually is complete bilayer formation around it. And if you look at the closer and downward structures we got, you'll see it's actually the vesicle yield is done, but 10 times better than in the presence of closures compared to just regular phosphorus. So first, just finding the right closure rate in lipid mixture is tricky. I'm not gonna lie, but we get a really interesting structure like this. Okay, so you might say, can you do this for different stuff, since I found it's difficult. It's difficult, but it's doable, right? To find the right combination. It can be done with proteins that arginine-rich protein, for instance. This is the protein sulfate closure rates. And this is PK, it's just a polymer-based closure rate. You can still use polymer-based things as well to form the structures. But they are quite different, but it's possible to use different processes. Okay, how do I know they form a bilayer instead of just typical assembly? I check the diffusion, I check the polarity experiments, I also do quantum experiments that I'm not going to talk about, but let's is the easy thing. We can look at the division. Let's look at that first. If you have any questions, I can share them. So here, right, I choose the region of interest and photovoltaic region. What I observed is that I had a quick recovery in the seconds. And if you, I calculate the fission coefficient, it was within a range that for sound and wave and fermion range is actually quite interesting. The value actually fell into these two values. So I have a mobile lipids around the closures. With our experimental result, I can confirm that I have a high rate. So this is quite interesting because I can use these systems for different things. For instance, if you have a closure rate, you can easily concentrate molecules in that closure rate. But if you have lipid molecules, right? If you want to concentrate something within a receptor, what you have to do is you have to use higher concentration. If you want something higher concentration within that receptor, you have to use tons of molecules. Conservation has an advantage over that. Of course, lipid vesicles provide another layer of protection of this content, right? Because when you have a closure rate, anything can enter and exit. So I did here I use RNA, USC, and I use some protein, USA, right? And I encapsulate this within the closure rate, and then I form the bilayer around it. Then I calculated the concentration between these droplets. I actually find quite interesting result. The starting concentration is 0.05 micromolar. I use very, very tiny amount of RNA and protein for this study. And when I calculate the concentration, what I observed is that almost 200 more times of the RNA, and I almost had 440 times of the protein I have between the droplets. So definitely RNA and protein were partitioning clonaries, and I were able to concentrate that in the droplets, right? And essentially it does have shortcomings of the vesicles. So here, if I only have vesicles, and I have the same concentration, 1.5 micromolar of RNA and protein, this is what they will look like. So here you can see the RNA in blue and protein in gray. So here you can't see any blue or gray. The reason is the concentration in and out is the same. And I check their histogram, or when you have the closure rate, when you calculate the drug vesicles using the closure rate, what you will observe that intensity ratio for RNA is almost 3 times higher than the outside. And for the protein, you almost have 100 times higher intensity for the droplets than outside. If you look at here, just the giant vesicle cells, the in and out, the intensity ratio is 1:1. Okay, so this is one thing, right? Closure rate is bringing to the table. The second advantage of closure rate can help with this system is just protecting the closure rate effects. So closure itself is vulnerable. So if you have an enzyme, let's say, it might actually just digest everything you have. Here, I have the protein sulfate closure rate. Without a membrane, I have the closure rate right here. If I add an enzyme like trypsin, in less than 2 minutes, trypsin is going to leave the peptides, and then you're gonna install the closure rate you have. The reason is then you have a formal chain line, right? if you decrease it from a chain length, it's related to the phase separation. Higher the chain length, higher propensity to phase separate. If you just dissolve, if you just digest these chains, you just have smaller molecules, they are not going to phase separate. Their propensity to phase separate will decrease. That's why when you use trypsin and cleave the bonds or artery, what's going to happen is you're going to dissolve the causality. That's not exactly what you're doing. However, if you around this compartments, when you add the trypsin molecule, it's going to start going to be able to lead the bonds of carbonate and splice over this you have in the covalent droplets, and it's going to be able to further and longer the molecules you have in the droplets. So this is quite interesting, and the first everybody says with phospholipids, why don't you use something simpler? You can definitely use something simpler. Here is an example. This is unpublished work. So you can actually use folic acid, for instance, to template membrane formation by oleic acid. So you have to again find the right combination of fluoride, and you can actually combine oleic acid also with PLPC at the same time. Because I also see that might be also helpful and beneficial. And you can have again this membrane formation for that as well. This is just a piece of data, and very usually more common, but hopefully so. Okay, then I try to show you that you can actually produce causal system membrane around different type of systems, not just one. It's a different type of models, and you can actually. I didn't show every data I have. Membrane fluidity. I show that you can actually have diffusion. If you have a diffusion, if you have a recovery, you have a bilayer, and I showed the third data for that. I also have polarization indication. You might see that here. You have different rates of fluorescence level that actually shows polarization. That's indication of lipid alignment. And you actually see the data for you can actually protect the interior components by the help of templates. And you can also concentrate biomolecules within templated vessels. So what did I show you today is actually everybody thinks that we need longer molecules, but that's not maybe the case, right? Because short, politically relevant molecules might be very helpful. They can improve the functionality of membrane compartments. In the first part of the talk, I mentioned that. And you can actually use covalent droplets as a template to template the lipid membrane formation. You can get higher yield doing that. And so combining condensates, combining covalent models and lipid membranes can actually help you to get more sophisticated with the cell and its internal systems. They do help each other. So most of the work today I talked was from my PhD. For my postdoc from MIT, I did more mammalian cell research, and now I have my small research group, and these are my students. They're following their first term solution right here. Very exciting moment for us. With that, I would like to thank you for your attention. I will be happy to answer any question you might have.
Okay, questions for Fatma? Yuck.
Yeah, really interesting talk. So I'm thinking liquid phase separation is also something
Yeah. Yeah, I didn't show the data, but there was studies showing that, for instance, if you change the salt concentration by just using simple arginine 10 mer ATP, you can be resistant up to 1 molar salt concentration. So I'm not saying definitely we will have the always ideal environment, but they're more stable than you would think. And again, lipids, for instance, might play a role, like protecting type of molecules. You have to make sure you have the correct charge and system for the systems, but I'm specifically focusing on type of phase separation. So there are other systems we just actually use solvation for phase separate. They are more complicated. So it's not just one type of system I mentioned. I'm just mentioning complex coagulation, but there are other types you can use. There is one type that you use PEG and dextran, for instance. You just have to have really high concentration of these large molecules to phase separate, and they are more resistant to condition. But if you think about cells, right, they're also not resistant to anything. So if you are looking for like that type of resistance, it's really difficult to find. But maybe we didn't need that much resistance, but sometimes, for instance, one period of time we need resistance to salt, right? And we can provide that with phase separation.
Zach.
Yeah. I think definitely charge plays a role. But charge actually plays a very important role. And the composition of lipids find that I am trying to find the potential. Okay, here. Lipids composition actually does affect significantly. These actually, I'm just changing simply the lipid composition, right? United form is the aggregate, but it's simply changing the one part of lipid. I'm also changing the transition. But charge definitely, even though we have a charging process, I wouldn't think like they are very charged. Especially the ones I'm showing you today, they are mostly have equal number of positive and negative charge. So if we did measure their zero potential, I don't think I have the data. My undergrad did that. So they actually slightly positively charged or negatively charged from the corner side. They are on the neutral side, and the surface of the closer is have enough charge. It's not strong enough. They can interact and they can form. So that's why you have to be careful with the balance. You cannot have too much charge. So there's a soft spot. I would like to actually model that and see that we can achieve critical. Because there's definitely a trend. I don't know how to put those into numbers or relation. It would be very interesting to do, but the charge actually is significant.
So let's go to Roy.
Yeah. Bilayer or monolayer. So I actually have a crunch experiment. Can you show that? Okay. I can't find my mark. Okay, now it's in the screen. Sorry about that. We have the crunch experiment. Okay. Trying to find. Okay, it's not here.
You could explain it in words.
So yeah, so I will explain that. So we have a crunching experiment. Basically, we put some thiocyanide, crunch the fluorescence we have the outside, and they are flipping over, right? They are flipping over rate for around 2 hours. So you wait for 2 hours and just measure the fluorescence afterwards. And if there's a decrease, around half decrease, like I mean, depends on the amount you have. So you actually see a decrease. That decrease actually tells you if you're flipping off, right? The quench force is actually you have the quench first, and it's moving over, so you actually see a decrease in your quenching. Tells you have actually a bilayer that you can have the flip motion. I would like to show you the data. Yeah. Yeah.
Me?
Different pH conditions for, definitely that's an interesting question. So if you change the pH, definitely you have to change everything about the system. And we choose the pH around 7.4 because it's relative to life. But it will be interesting to check how the pH affected how these compartments react to that.
So I have a question, which is, so we're interested in concentrating things so that reactants can react together and produce something relevant to prebiotic chemistry. And have you thought about going to the next step and putting in reactants in solution that can concentrate in your coacevates and then do something that is prebiotically relevant? Have you thought about that?
That's a very interesting question. There's people in my first group, they did RNA polymerization in this droplets, and they get concentrated. They show that without concentration effects, they weren't able to get any reaction in the pulp. When they had the closures, they were able to get the reaction going. So they actually have a nice.
And how many ribonucleotides did they eliminate?
Yeah, I think I remember like 3 or 4 or 5. I think they did more. It's a 2019 paper, but they were able to definitely do that. And another group, Boateng, also was able to do this correlation. That's the direction the field is going, right? These systems are interesting. What can they do?
Right. And then the other question I had, or really it's just a comment really, that maybe you can expand upon.
Sure.
So while we were talking before this talk, you said that some of this could be medically relevant.
Yes.
Like Alzheimer's, and maybe there's other diseases,
yes.
but tell us about condensates in disease and how this could be relevant.
Yeah, I can show my data actually. So what you see here is delivered N protein. N protein is the encapsulation protein that can encapsulate anything that's related to this for COVID. Right? It's called that encapsulates virus. So if you just deliver that, right? This is just a simple example, right? You have the molecules just condensated in the system, and they're interacting with ER. Here you see ER. It's highlighted in magenta. So basically, they form these clusters on ER, and ER plays an important role for virus replication. So that's another way people are looking. For instance,
you can suppress the virus replication. Is that what you're saying?
I mean, I'm just saying like any protein might just condense on the ER, and that's how they can replicate. That's one way, and that's actually what I'm showing here. But a lot of people work with prions and different molecules, ALS and Alzheimer's. There's this future formation. So when you look at this, they show that when you change the physicochemical properties, then they cannot form any separation, and it is like nature forms fibrils. It does cause this disease state as well. So again, the fundamental physical understanding the system can apply all those different diseases, things, and I mean even virus replication.
Any other questions? Any questions online? If not, let's thank Fatma once again.